Hello everyone. Thank you for joining us today on the webinar on RNA read alignment using Avadis NGS. As most of you might be aware, Avadis NGS is a next-gen sequencing product which has been in the market for a year and a half now. The USP of the product is that it makes analysis simple to carry out for biologists. It is developed on the same platform as GeneSpring. So those of you who are using GeneSpring for microarray analysis will find the look and feel of the tool very similar. Avadis NGS, right from the first edition, supported import of aligned files which were in SAM, BAM or Illumina-specific ELAN formats. The first version of the product had workflows for ChIP-seq analysis, RNA-Seq analysis and DNA-Seq analysis. Small RNA analysis was added one release later. In addition, we also provided alignment support for small RNA reads and DNA reads. The upcoming version of Avadis NGS will feature RNA read alignment also. In this webinar, we'll go through the details of the RNA read alignment workflow. The outline of today's webinar is as follows. We will first take a brief look at DNA read alignment, just to place in context the difficulty of aligning RNA reads. After that, we'll describe the origin of spliced reads in RNA alignment. Then we'll move on to the different ways in which we can align RNA reads. For instance, we could align them against known transcripts alone, or we could try to detect novel splices involving known exons. In addition, we could align against the genome itself and try to identify novel exons. We'll show you how support for these different types of alignment is included in the tool. Then we'll show how to create an RNA alignment experiment in Avadis NGS and how to choose the different options with which to carry out the alignment. And after that, with a single click, you can create an RNA analysis experiment from the output of the RNA alignment workflow. And in the RNA analysis experiment, you can carry out all the traditional analysis tools like quantification, differential expression, differential splicing, go and pathway analysis, and other downstream analysis. So let's start off with DNA read alignment. In DNA read alignment, we take the reads which have been output by the sequencer, we match them against the genome, so we have contiguous matches to substrings of the genome. So DNA alignment looks like this in the genome browser. What we see out here is a coverage track at the top. This is a resequencing experiment, so you can see the coverage is uniform and quite high in the 130s. Below that, you have the reference sequence. The reference sequence is only color coded out here because we are zoomed out at a certain level. And below the reference sequence, we see the individual reads. You can see that most of the reads match perfectly to the reference sequence, though you do have some mismatches out here, and you also have some bases which have been soft clipped by the quality control. But overall, the read sequences are substantially similar to the genome sequence, which makes the DNA alignment problem a little bit similar than the RNA alignment problem. Let's now take a look at the RNA read alignment problem. Suppose we have a transcript with 2 exons, and the 2 exons are shown separated by a distance on the genome out here. When the mRNA gets expressed, the intron gets spliced out, so what we have is a transcript molecule with the 2 exons adjacent to each other. Let us say that when this particular transcript is fragmented as part of the sequencing protocol, there are 3 reads that come out of it. Read R1, which falls entirely within the first exon, read R3, which falls entirely within the second exon, and read R2, where part of it comes from the first exon and the second part comes from the second exon. So when we align these reads against the genome, R1 will have a contiguous match against the genome, it's a single match. Similarly, R3 also has a single match against the genome, but R2, the first part of R2 aligns to one genomic location, and the second part of R2 aligns to a second genomic location. So this is what we call a spliced read, and the 2 different parts are connected by a dotted line in this particular picture. So to summarize, reads R1 and R3 have contiguous matches against the genome and what we call as unspliced matches, whereas read R2 has a spliced match, which is typically represented by a figure of the form, which indicates the length of the matched locations and the length of the intron that it is spanning. So given a read, trying to identify the splices that it has against the genome is computationally a difficult problem. For instance, suppose we take the read out here, we'll have to figure out how many spliced parts it has and what are the genomic locations of each one of those spliced parts. So research papers talking about alignment of cDNA sequences against genome sequences have been published since the 90s, and they continue to be published today because the problem of aligning a cDNA sequence against a genome sequence continues to remain a difficult problem. And as read lengths get longer, the same problems that were encountered in aligning cDNA sequences against genome sequences surface when you're trying to align RNA-seq reads. So let's take a look at the different RNA read alignment approaches. We could try to simply align the read as a contiguous match against the genome. That is, we could try to do unspliced alignments. This would work effectively if the read sizes were small. For instance, if the reads were only 30 base pairs or 35 base pairs long, as they were 2 years ago, then the chance of the read spanning an exon boundary would be very low, so the read could be mapped against the genome, and that could form the basis for quantification. However, read lengths have been getting longer, and read lengths of 140 to 150 length are quite standard these days. Now, given the exon length distribution for the human genome as shown in this picture, the median exon length is 140 bases. So the moment you have reads of length 100, then most of these reads are going to span across exon boundaries. So when you try to align against the genome, most of these reads will not form single match against the genome. For instance, with 100 base pair reads, we can show that only 38% of the genes in the human genome will be quantified accurately if you simply do unspliced genomic matches. Since unspliced alignment against the genome is not an option, the next thing one can consider is alignment against the transcriptome. For instance, suppose we have a genomic sequence out here containing a transcript with 3 exons, which are shown in yellow. Then we could construct the transcript sequence, which is the concatenation of the 3 exons, and align the reads only against these transcript sequences instead of aligning against the genome sequence. Now the read will have a contiguous match in the transcriptome, so we can reuse the same tools that we used for genomic alignment. That is, we could use unspliced read alignment tools, but instead of aligning against the genome, we could align against the transcriptome. And once we align against the transcriptome, with the knowledge of the exon-intron structure of the transcript, we could translate the reads into genomic coordinates. For instance, the first read out here is falling entirely within exon 1, so it would form an unspliced read when translated into genomic coordinates. However, reads 2 and 3, because they straddle exon boundaries, we can translate them into genomic coordinates, but we would create unspliced read in that process. Whereas read 4 would be an unspliced read when translated into genomic coordinates. So if the transcript model is fairly complete, that is, if you are working with well-studied organisms whose annotations have stabilized over the course of the years, then aligning just against the transcriptome is a great choice for carrying out RNA-Seq analysis. However, suppose knowledge of the transcript model is incomplete. For instance, suppose we are aware of one particular form of a transcript which involves 3 exons, but there happens to be another alternate form which involves only the 2 exons, the first exon and the third exon. Now, given a transcript of the second form, if we derive reads from this particular transcript, and the read happens to straddle this exon boundary, this read will not align in a contiguous manner against transcript 1, because part of the read comes from exon 1 and part of the read comes from exon 3. So this read cannot be aligned using traditional DNA alignment tools against the transcriptome. Some specialized tool is needed, and that tool would have to break this particular read into 2 parts and try to figure out for each part what is the possible exon from which it arose. So this is what we call as aligning against the transcriptome, but detecting novel splice forms. And Avadis NGS supports this particular feature. When carrying out alignment in Avadis NGS, you could indicate that alignment needs to be carried out only against the known transcripts, or you could also say that alignment needs to be carried out against transcripts, but novel splice junctions need to be detected. In this particular view, we are showing a simulated data set. The data was simulated using the transcript model shown at the bottom track. However, the data is being aligned against the transcript model shown just above that in a track called aligned transcripts. We have carried out alignment in 3 different manners. We have carried out alignment against just the transcriptome. We have carried out alignment against the transcriptome with the novel splices option on, and we also carried out alignment against the transcriptome and the genome. Now consider the reads which arise from the transcript shown at the bottom track. Suppose we have reads which span this particular intron. In other words, part of the read comes from exon number 3 and part of the read comes from exon number 4. When we try to align those reads against the transcriptome, they will not match because the transcriptome that we will be aligning against has the form where there is a novel exon in between. So these reads will not match, and that is indicated by the empty space that we see out here. And we can also see the odd-shaped coverage because the reads that arise out of the exon boundary between 3 and 4 One part of the read will be towards the end of exon 4 and the other part of the read will be towards the first part of exon 3. And since these reads don't align against the transcriptome, the coverage in both of those locations is very low. So we see an odd-shaped coverage on these two exons. But if we carry out an alignment with the option to detect novel splices, then we can suddenly see that all of these reads do indeed get aligned, and we can see that the coverage is restored to a more meaningful looking coverage pattern. Similarly, let us consider the case where there is an exon that we don't know about in the annotation. In other words, the transcript model that we have in our annotations has only 5 exons, whereas there exists a transcript form which has 6 exons. It has this additional third exon out here. Now, if reads are expressed from this particular transcript, then the reads which contain bases from the third exon, those reads will not align against the transcript model. They will only align when we align the reads against the genome also. And we can see that Avadis NGS handles those reads also fairly well, because we can see that there's a coverage out here which corresponds to the exon which is missing in the annotation. Similarly, suppose we have the annotation containing only the transcript model shown out here, where both of the exons are fairly small, but in reality, there's an alternate transcript which has an exon extension. In other words, first exon out here is much longer than what is in annotation. Then all of the reads which arise from this particular part, those reads will not align against the transcriptome, but they will align against the genome. And we can see that Avadis NGS detects those alignments. In addition, it also detects spliced alignments involving the first 2 exons. In a similar manner, 2 alternate transcripts might differ in terms of intron retention. So the annotation that we have in the tool might have 2 introns, whereas in reality there might be a splice form which has only 2 exons and it incorporates the intron from the first form. Again, when reads arise from this particular transcript model, those reads will align only against the genome, and we can see that Avadis NGS handles this case also. So all of the common cases arising from incomplete annotation in the presence of extra exons, exon extension events, intron retention events, all of those events are handled by the Avadis NGS aligner. Finally, you might have some entirely new genes which are not present in the transcript model at all. For instance, here is a gene which has 2 alternate transcripts, and suppose these transcript structures are not present in the annotation. Avadis NGS still manages to detect the presence of those reads. It aligns against the genome, it finds enriched regions, it checks the enriched regions for the presence of appropriate splice signals and aligns reads using the novel exons detected in that manner. So one can see 3 different novel exons out here, and one can see perfect slices aligning with the boundaries of the exons. So now that we know the capabilities of the aligner, let's take a look at how the alignment experiment can be created. We start by creating a new experiment and choose the experiment type. As mentioned earlier, we have 4 different analysis experiments: ChIP-seq, DNA variant analysis, RNA, and small RNA. And now we have 3 different alignment experiments: a DNA alignment, from which we can create ChIP-seq or DNA-seq analysis experiments; a small RNA alignment, from which we can create a small RNA analysis experiment; and the newly introduced RNA alignment. Once we choose the alignment type, we specify the metadata for the experiment. We choose the organism and build against which we want to align the read. We choose the sequencing platform, and we have support for all the major sequencing platforms. After that, we choose the library layout. We could be aligning single reads, paired reads, made paired, or even directional single-end reads. Next, we choose the FASTQ files which are output by the sequencer. Here we have an option of merging multiple files by providing the same sample name. In this case, there were 5 different FASTQ files, all of which correspond to the HBR sample, and another 5 files which correspond to the UHR sample. So all the reads on the first 5 files will be combined into a single sample called HBR, and all the reads from the latter 5 files will be combined into a single sample called UHR. Once the reads are read by the importer, we can do read level QC. Avadis NGS provides a very comprehensive set of QC metrics. One can look at how the base quality is changing with the position in the read. In this particular case, we can see that the quality in the first position is close to 39, the median quality, and the median quality continues to remain the same all the way till the end of the read, which is 50 base pairs long. But the mean quality does decrease a little bit from 37.5 to 34. We also provide a base composition by position plot. Here indicates that within the first 10 bases, there seem to be preference for certain bases at different locations. However, after a while you can see that the GC composition stabilizes at the GC percentage of the genome. Then we have the base quality distribution plot, where we count for each quality the number of bases with that particular quality in the sample. You can see that here the quality is excellent, though we do have a few bases which are of quality 2. The base quality summarized across the length of a read is the average read quality metric. So we can plot the read quality distribution, and it does look similar to the base quality distribution. Based on all of these QC views, one can choose the appropriate filtering options to be carried out later on. Then we can start the actual RNA alignment. And when carrying out the alignment, as we discussed earlier, we have 3 options for doing the alignment. If we believe that the transcript model is quite complete, or if you're interested only in the known transcripts, one could choose to align against just the transcriptome. If you believe that the transcript model is fairly complete, but there do exist alternate isoforms involving the same exons, one can choose the second option, which is transcriptome with novel splices. However, if you believe that there might be novel exons or novel genes in that particular annotation, one can choose the last option, where the reads will be aligned against the transcriptome also, and reads which do not align against the transcriptome, they'll be aligned against the genome, and the best alignment overall will be output. One should be aware that each one of these options takes a little bit more time, so the transcriptome only alignment is quite fast. The one with novel splices will take a little longer, and the last option will require more time depending on how many unaligned reads are there. In addition, you will need a 4 GB RAM machine to carry out alignment with the last option. And then we can choose the transcript model against which we want to carry out the alignment. On the Avadis NGS server, we provide annotations from multiple sources, such as REF-seq, UCSC, and Ensemble. One can choose the source and the particular version of the transcript model that we want to align against. We can then set the alignment criteria. Usually we set the minimum percent identity to 90. However, if you believe that there are a lot of sequencing errors in this particular sample, we could lower the identity percentage. Similarly, depending on the expected number of indels in this particular sample, we can set the percentage of gaps. The default value is 10. If we are trimming the reads based on quality, the trimmed read might become very small, so we could set a limit on what is the minimum match length and the number of multiple matches that need to be reported. Then we can set the trimming parameters. If the reads have adapters present at either end of the read, one can choose fixed trimming to remove those adapter sequences. One can also trim based on the quality to get rid of the bases which have low quality, typically towards the end of the read. One can also carry out trimming after alignment is carried out. So if there are a lot of mismatches at the end of the read, we could choose to remove that segment from the match record. After we set all of these parameters, we can carry out alignment. And as you can see from the navigator shown out here, one can carry out alignment multiple times. In this case, alignment was carried out thrice, once against the transcriptome only, once with the transcriptome with novel splices, and once against the transcriptome and genome. Even with the same options, one can carry out alignment with different percent identity and gap parameters. And after the alignment result is generated, one can take a look at the alignment report, which indicates the total number of reads that were processed, the number of reads that were aligned, and it also qualifies the alignment as how many were aligned against just the transcriptome, how many reads involved novel splices, how many of them involve the genome, etc. Once we are satisfied with the alignment results, one can choose the alignment output and create an RNA-Seq analysis experiment from the alignment results. The details of the RNA-Seq analysis experiment have been covered in a different webinar, but to briefly summarize, we can carry out quantification of all the samples. We can normalize the quantification result using DE-seq or TMM. One can then set up interpretations and compute differential expression results. One can also carry out differential splicing analysis, and the gene view within Avadis NGS will help verify differential splicing results. Genes which have been identified as important through the differential expression or differential splicing analysis can then be used to carry out GO and pathway analysis. That brings us towards the end of the webinar. If you would like to try out Avadis NGS, please visit our website at www.avadis-ngs.com. We provide installers for all platforms, Windows, Linux, and Mac, 32-bit and 64-bit versions of these installers. We also provide multiple demo data sets to make it easy for you to get started with the tool. As I mentioned earlier, we provide comprehensive annotations. We take gene and transcript models from UCSC, Ensemble, and REF-seq. We make them available on our annotation server, so you can download them with a single click. We also provide annotations for a whole lot of other organisms And if the organism of your choice is not there on our annotation server, you can always create those annotations within the tool itself, starting from GenBank files. We also provide pathway interaction databases for human, mouse, rat, Drosophila, zebrafish, yeast, C. elegans, and Arabidopsis. The Avadis NGS tool's functionality can be extended through custom scripts that we can provide if there is a need. Scripts which have been found useful by multiple customers are put up on scripts library on our website. Thank you.